So when I actually run this, it prints those 3 samples that I had listed earlier. Like we mentioned before, so you always start with your target rule, and you can name these whatever you want. So the syntax is rule, and then it'll say, then you type in the name of your rule. So I use all, you could use target, whatever you want. So the input to the target rule, again, is the last thing that you want the pipeline to produce. Here, I want a directory called differential expression, and I want a gene count matrix in a CSV format as my output. So moving on to the second rule. So this is actually the first rule that's going to be run, and this is the align high sat rule. So again, these SnakeMake rules are broken down into 3 general categories. You have input files, output files, and then the shell or any sort of command that you want it to actually run. This log and threads are optional. I just have them in there for completeness. So for my inputs, I've got the FASTQ1 file. Here I'm just pulling the data path. The nice thing about SnakeMake is it allows you to use what's called a wildcard. So instead of manually typing in all of these samples to have 96 samples, you can just use a wildcard. And so SnakeMake will pull this from the config file. It'll know that I want it to run this using all the samples that I have specified, and it'll run this pipeline for all 96, and you only have to specify 1 rule. So I've got the sample as the wildcard, which is marked by these curly braces. And then the last is the extension that I had them specify as well. Same thing with FASTQ, the second file, same exact syntax. And then I'm also specifying as an input this HiSat2 index. And here, this is just an expansion code. So there are like, I believe 9 files for that HiSat2 index. And so it's just saying for every single one of them that ends in .ht2 to include those as inputs rather than typing out all 9. The output of my align HiSat rule is going to be a BAM file. So it's going to be created in this align HiSat2 sample directory, again, using a sample as a wildcard. So it'll fill that in with the sample name and then a BAM file. Then I'm logging the alignment summary here, again, with the wildcard. I'm specifying the number of threads that I had set to 8. And then this is the actual command that I'm going to be running, which is the shell script. So I've got HiSat2 specifying the log, -p for specifying the threads. Here, this -x for specifying the HiSat index, and then -1, -2 for the FASTQ file inputs. And I'm piping all of that into a SAM tools sort, specifying the number of threads and specifying an output to convert that SAM to a BAM file. And that's all in 1 rule. And I know that's probably a lot to take in. So I've added 1 more slide that actually makes it a little bit more human readable. So when you actually run this pipeline, you can use a dry run. So instead of just running it all and having it crash and burn your first time when you're trying to figure out all the syntax, you can use a dry run where it'll just print the statements. And so this is a dry run of my first rule. And so as you can see, it's filled in the sample wildcards with exp1 here. It's listed all of the different index files ending with ht2 for the HiSat index. It's got my output as exp1.bam. It's got my wildcard listed as exp1, and it's got a job ID there. And then down here at the bottom, this is the actual command. So it's got the inputs and it feeds it into that command that I had. So again, it's just piping everything into the SAM tools and finishing up converting it to a BAM file. And on my GitHub, as you can see, I'll have all the other files or all the other rules written in that SnakeMake file. But for the sake of time, I'm not going to go through every single one of those, but they all follow the same syntax. And you can create a rule for essentially anything you can think of. Doesn't have to be Python, any sort of software works. And it's very easy to scale, very easy to add more samples, and very easy to thread on an HPC. And that's all I had. Thank you, and I'll be happy to take any questions.
Thank you, Will. Yes, there is actually 1 question in the chat. You mentioned how the thread number requested correspond to the SBash directives for the number of CPUs, etc.
Oh, sorry, I'm pulling up the chat right now. Sorry, can you say that 1 more time? What about the SBash?
That if you could mention how the thread number requested corresponds to the SBash directives for the number of CPUs.
Yeah, so you kind of have to, the way I have it set up in a Slurm script right now is that I am activating the Conda environment and I'm actually specifying that Slurm script, how many threads I want CPUs. So you kind of have to do it in both places, both in the config file. But if you just ran that on your laptop, you just need the config file. But if you're running it on an HPC, you have to kind of manually create a Slurm script as well. I'm sure there's a better way of doing that, but I input it in both places.
Okay, thank you, Will. There are a couple of questions there in the chat that if you feel free to just go and try to answer them. We're gonna give now room for Dennis to give his presentation.
Go off mute here. Okay, we can see my screen. Yeah, so I'm Dennis, and today I'll be giving a higher level overview of what workflow languages are, what purposes they seek to accomplish. So we'll just kind of dove into a specific example, but this will be more general.
Dennis, quick thing, you are in the presenter mode. I don't know if you meant to have that.
I'm in the presenter mode.
You are
yes,
in the...
I see. So let me share my other screen. Thank you. Let's do this. Better? Now you can see my Slack channel.
Yes.
Okay. Okay, there we go.
Got it. Thank you.
Now I'm too far ahead. So yeah, so what is a workflow language and why use one? In many scientific fields, such as bioinformatics, medical imaging, astronomy, large quantities of data need to be analyzed regularly to produce meaningful results. And these results generally require large scale and repetitive processes involving many different tools or software over several steps chained together to go from raw data to a final result. And the chaining of these individual steps in a pipeline is commonly referred to as workflow. So it's very time consuming to run data through each of these individual steps, and doing them meaningfully at scale isn't possible if you have to continue constant manual intervention. So historically, to solve this problem, researchers and software engineers have baked together custom workflows using either Bash or Python to allow the computer to chain these steps together along with database triggers to run subsequent steps. But these are often local to a group, and they even differ from group to group at an institution or a department. This becomes incredibly system dependent very quickly, as we often try to accommodate for our local infrastructure. So we mentioned Slurm, our scheduler could be a part of that, our database, our environment variables, whatever we use that's tied to our system is non-transferable. So they make them inflexible and not shareable, really. So workflow languages alleviate this problem by allowing these workflows to be expressed formally, as you saw with SnakeMake. They provide infrastructure to set up, execute, and monitor the workflows. There are over 100 different workflow descriptions to choose from, but from my experience in bioinformatics, the three most common are WDL or WDL, CWL, and SnakeMake. Those seem to be the ones that people use the most. And here I have a little picture. So this is an oversimplified example of a bioinformatics workflow that we run at St. Jude. In this workflow, there are 10 steps or bubbles, and the order of these steps is exemplified by the arrows. Each major step has substeps of 4 to 5 scripts. So for example, mapping of raw data contains splitting the data, aligning the data to a reference genome, sorting and merging the data, and then duplicate marking. So there are actually 4 requiring different programs and different invocations of software. So all in all, there are roughly 50 separate command line executions required for a single sample to pass through this entire workflow, and that is only for a single sample. So we might process 20 samples in a batch, producing 100,000 files. Each of those files has to go through these steps, and that's just simply not doable by yourself. So this is kind of a breakdown of the general premise of what a workflow language might look like. So I won't go into too much detail here, but generally they're broken down into tools or steps. So individual steps that need to be run that describe the program, the command line arguments and the inputs, and then these are chained together in a workflow that's executed sequentially or semi-sequentially if some steps are conditional. So here I have tools on the left with the workflow on the right. The general premise is that, in the top left, you see split. It has the command to run, which is just the Linux split command, split it by 2 million lines. The input to that is a UBAM and it produces an array of UBAMs as output. And then if we look through, we can see that each of these steps takes a BAM or a UBAM as input, and it produces some other type of BAM as output. So if you ran those steps individually, that's what you get. And then on the right, we have the workflow. And here you see the only inputs are, a single UBAM and a reference. And then if we look at the middle section of the script, under align, we see the inputs are they're from the output of split, and it's an array of UBAMs. So it's going to take the inputs of the previous step, the outputs of the previous step as the input to the current step, and eventually you'll get your final result. This isn't this is just a general overview of what this might look like. So motivation for using workflow languages. We want to make computational methods reproducible, portable, maintainable, and shareable. So I'll talk on reproducibility a bit. There is a reproducibility crisis in research. In 2016, Nature did a survey. This is this top left picture we have of 1,500 researchers on reproducibility in research, and 52% of those surveyed said there was a significant reproducibility crisis, meaning they couldn't take someone else's work and reproduce it locally, while 90% said there was at least a slight reproducibility crisis. Furthermore, 34% don't even have procedures in place to ensure reproducibility from run to run or experiment to experiment, depending on what kind of work you do. These numbers come from a variety of fields, not limited to computational methods, but if you've ever worked on a research project, you've almost certainly felt the pain of taking somebody else's code and trying to run it end to end to produce results. And there are many things that can contribute to the lack of reproducibility, but I'll just talk about a couple. I think the biggest one is inconsistent software versioning. So software version, changes often, and they don't create huge noticeable changes, but if I use version 1 of a program and I send it to you and you try to do the same thing with version 2, you kind of get, over the course of a workflow, what I call the telephone effect. So if you've ever played telephone, it's where you whisper a sentence in someone's ear, and after the first person, it's only slightly different or maybe the same, and then the second person, it's a little bit more different, and then by the 5th person, it's very different. The 10th person, it's unrecognizable. It's a different sentence. So this same challenge can happen with data. Subtle differences are exacerbated over the course of a pipeline. So if my step 1 changes data in any type of way at the beginning of a workflow, the subsequent steps are going to be affected by this. Runtime environments also can cause lack of reproducibility. So changes in OS can contribute to this. An example I have from that is we upgraded OS versions from Red Hat 6 to Red Hat 7, and the order of the Linux command ls, it flipped. So it used to do ascending order, and then it started doing descending order for alphabets or numbers. And we had a pipeline that was affected by that. And then the last one is installation of software. Sometimes installing software can be challenging, especially if it's very old. So workflow languages seek to solve this crisis by providing researchers a well-defined set of steps with specifically defined tool versions in a given OS. And the best way to do this is via containerization. So if you're not familiar with containerization, it uses OS level virtualization to deliver software in packages. Containers are isolated from one another and bundle their own software libraries and configuration files, and they can communicate through well-defined channels. And this level of control allows us to transfer exact replicas of our local whatever environment we have set up to another person. So they just execute the container and they get the exact system that we use to run the program. And workflow languages handle containerization, which is excellent. So they're also portable. When I speak about portability, I don't mean in the sense of place to place per se. What I'm referring to here is system to system. So with the workflow language, the design should be, if you follow what I just talked about with containerization, we should be able to run the same workflow on a laptop, a small cluster, or an infinitely scaling cloud. One caveat here is that tools sometimes, they require more memory than a laptop might have. So, the computer's your limiting factor here, but not the workflow language. So because most workflow languages can be executed in local or batch modes, the execution of the start of the workflow and the configuration file passed, may be different in each of these environments, but the results, they should be the same. Will mentioned using like a Slurm script to execute the SnakeMake. So that might be required, but the results should be the same. Maintainability. So line count of code. This is a software engineering thing, but one thing that workflow languages can tremendously reduce is the line count of code. Since infrastructure definition of pipelines and chaining of steps requires external custom definitions for Bash or Python scripts, you end up with a tremendous line count when it's all said and done. One researcher from the Broad Institute reduced his line count by 73%, switching to workflow languages. And that makes it easier to test. So we need to be able to test all of our environment setup, execute. So subtle changes aren't going to break our system, and share and run in the cloud. So when you're, responsible for your own infrastructure, you have to maintain the setup and execution of the scripts, like I said, and make sure that they're creating the workflows properly. For large-scale research institutions like ours, this portion has tremendous overhead. This is something we see every time we do a software upgrade or introduce a new portion of our system. We have tremendous overhead even in testing our setup. So the last and potential most important piece that I love about workflow languages is basic unit testing. Unit testing seeks to test the fundamental block of code and ensures that the inputs and outputs are concordant. These tests are important because they validate your code. So if you update the workflow language version, the tool version, the software version of the tool that you're using, or actually make code changes to the workflow itself, you can do an end-to-end test. So the way I do this, I'm not a representative of GitHub or Travis CI, but you have a GitHub repo, you have inputs for a specific program that you want to run, and they're mock inputs, they're small, and you run the code manually, you generate outputs, and then you run the workflow, and you test that the workflow outputs are the same as the manual outputs that you've run for a small, case. And then you push this up to GitHub, you integrate Travis with GitHub, Travis CI with GitHub. And then if you upgrade your software, say you make a new container with a newer version of software, you go from version 1 to version 2, you can just change the workflow locally. You can add the new container name to your workflow, push it up there, and then Travis will execute all of the code, making sure that it runs and generates the same outputs that it did previously. And I think this is really fundamental because this is what's going to tell somebody if, them pulling the tool, locally is going to work. So they can pull down your workflow, they can run your unit tests, and they can say, hey, I at least know that I can run this workflow now. And I can pass my inputs to it and check it out. And lastly, it's shareable. So, just doubling down on this, GitHub plus containerization makes it incredibly easy for me to give you my workflows. If you want to reproduce my work, if you want to try to run this on your own data, you could run a Git clone of, my GitHub repo, and then you could run the unit test to make sure that the software is functional and that you have everything working. And this is what it seeks to solve, is the transferability, the portability issue, and it doesn't require, another institution to spin up their own way to scale massive size production workloads to process data. So in conclusion, workflow systems should simplify your workflow. They should make scientific workflows reproducible, portable, maintainable, and shareable. And big takeaway, we should always design with the end user in mind. So we should design with tests and for shareability. And some references here.
Perfect. Thank you, Dennis. There is a really nice conversations going on in the chat.
Yes.
If anyone has any questions right now that want to ask directly to Dennis, you can unmute yourself and just ask, please. It's definitely a great conversation going on in the chat. One of the things that they're asking, Dennis, is that if you can please add the notes examples and how reproducibility can be affected by changes to the system, different version changes in iOS.
Cool. Okay.
Okay, with that, so while Matthew is getting ready for his presentation, I just need to find my notes here somewhere. Where do they go? There it is. So next speaker is Matthew Garcia. Matthew is a postdoc research associate at the University of Wisconsin-Madison. He has been focused, his work has been focused in assessments of climate patterns and changes in the upper Midwest in the US. And currently his postdoc work is focused on multi-stage modeling of forest insect outbreaks in migration in Eastern Canada. And so with that, Matthew.
Thank you. While I bring this up here, one thing that I wanted to bring up, I'm really appreciative of the bioinformatics orientation of the two presentations that you guys have given already. And I want to thank you guys for covering a couple of the basic things, especially things like reproducibility and portability. Those are a couple of points that I am going to bring up in my work as well. Earth science data is a growing area of reproducibility, or excuse me, of big data. And my work in my dissertation covered a couple of topics. Let's see here. So can you see my slides there?
Yes, we do.
Okay, great. So my dissertation topic, I wanted to look at the influences of climate on phenology in forests in the upper Midwest. Phenology is the seasonal cycle of biological events, things like leafing out on the trees, leaves fully opening in the fall, then turning to different colors, falling off the trees. There are similar processes in evergreen trees. Evergreen trees do not keep their needles forever. Their needles just cycle through on such a slow basis that that the tree itself stays green, but the needles are constantly renewed over a long period. So my dissertation study area covered this about 200,000 square kilometers using satellite images from Landsat. If you're familiar with that program, that produces a lot of the aerial, or excuse me, satellite imagery that we use over North America and a good chunk of the world. There are a couple of other satellites that I've worked with as well. And then I use ground-based data for the weather and the climate information. And when I started my dissertation back in 2011, restarted on this actual specific path in 2013, there were a couple of considerations that I had. My advisor and I were deeply committed to the idea of open source, open data, open publications, things like that. And so we wanted to make sure that we were doing all of our programming and analyses in ways that we could pass on to others, that would be transferable to other locations. If somebody wanted to pick up this procedure and look at a different area of the country, different area of the world entirely, they should be able to do that and, you know, freely using the tools and the workflows that we develop. The other, one of the other considerations was that we're very lucky here at the University of Wisconsin-Madison to have our own Center for High Throughput Computing. And I want to thank Christina Cook for recruiting me to give this presentation today. She works there at the CHTC on campus, and we've known each other for many years now and worked together on different aspects of the projects that I'm trying to do using those resources. So CHTC works on basically highly distributed workflows and applications. There are high performance computing clusters available within CHTC. I use those on my more recent work, but they're not part of what I did during my dissertation. My dissertation was oriented primarily on highly distributed single thread processes to get through a workflow that looks like this. So this was actually the first part of my dissertation. This was generating a climate-based or a climate-oriented data set for the area around Western Lake Superior. We eventually published this. In fact, it was the first look at climate warming in this area. We found that there was just about a degree of climate warming over the last 35 years or so. And so we published that, and it has been since reinforced with a couple of more recent results from other groups. Going through this flowchart here, this is kind of how I build up my workflows. I'm not going to get into showing too many details of individual pieces of the workflow, especially the code, although I will show one piece. But you can see there, the narrow boxes that you see on the left there, those are basically single processor operations, processing the original data set, getting through to where we need some more distributed work. And those are the wider boxes there. Those would be like if you were looking at a DAG, and thank you guys for introducing the conceptual visualizations of things like DAGs and the workflows in the presentations before me. It would be like a diamond DAG where you get down to you split from one chain to as many as you need. But in this case, I had 33 years of data, and I could process each year separately without dependence on the others at a certain point. And so that's where you see the generating daily weather maps with the call out box for how that went using spatial interpolation methods. Moving on from there was a much more complicated process, and that is where my use of HTCondor moved into using the DAG manager application that's written here at UW-Madison. It's called DAGMan. And I built eventually, like I said, we published that. I'm working on a piece of my website that actually has a lot of the background to that and a lot of the results and a link to the GitHub repository where all of my workflow and my processing code is all stored and a lot of the results as well. So moving back to that, what we have here is this is the HTCondor, the basic unit of processing. So there's a submit file, which works with the HTCondor system to match resource requests with what's available in the distributed grid computing system. And inside that are the data pieces, the Python, the modules that I need, things like that, and the scripts that actually do the analyses that I'm looking for. When I string a lot of those together, we can work in the distributed acyclic graph mode, and there's a manager for that written on top of HTCondor here. excuse me, which can string together any number of Condor submissions in whatever order you need in order to get to your results. And in some cases that gets so complicated that I write a generator to produce my DAG workflows. And that generator is usually a pattern-based Python script. And I can show you a quick example of that. This is basically a short Python script, you know, less than 100 lines that produced a DAG to run what I'm about to show you next, little animation that I built using the same DAG script actually, that puts things in the right pattern. So you see here, we talk about parents and children in the DAG process. And so one job may produce 2 children in this case, or 2 children may need to be, or excuse me, 2 jobs may need to be completed before one next child job is run. And so we actually have a procedure that I built up that takes care of calculating a number of derived variables from the basics that were generated in the step before. We needed to calculate derived 72 derived variables that had to do with climatological information. And some of them, most of them were calculated on a rolling basis. So they had to cross the year boundary. Calendar year doesn't mean anything really. It's more arbitrary. So a lot of those variables were calculated across the boundary between December and January on a rolling basis, but with the constraint that we couldn't have multiple analyses running to produce the results in the same file at the same time, because files get locked when they are being operated on by Python. And so what we needed was a way to run as many things as possible at the same time in order to save time overall, but not interfere with each other. And so that is this step here. And I can show you a quick animation of how that worked. This is what I called the Flooddag. And it'll show you in a moment how it operates. But basically the idea is we have these sequential and rolling calculations of derived variables, but we can't interfere with file access between 2 different operations that are going on at the same time. And so each calculation spawns 2 child calculations, and then those move through. And you can see here, this would be, each row would be 1 year of data, and it's calculating these different derived variables in sequence, and then moving down through the years of the data set until they're all completed. And so that was one way that I had approached this piece of the puzzle, rolling through with a pattern-oriented workflow that I could code up with a generator and then run on our HTCondor system. Going back from there, so that piece of our research is published. It's built so that I can update it every couple of years. In fact, I have an update that I need to run to bring the climatology data up to the end of 2020. And there are, since all the pieces are broken down into their most fundamental units, I can just run a couple of years and update the data set as it already exists without having to go right back to the beginning and rerun everything. The second part of my dissertation was oriented on applying that climatology data to see what types of variables, things like temperature and precipitation over different periods of time affect what we see in our satellite images at any given time of year. And so the data that you might get at a specific location in the forest near Lake Superior might look like what you see in the middle there. And this particular variable is a proxy for how green is the vegetation in that pixel. And so it's not very green at the beginning of the year, day of year there down at the bottom. So from the transition from winter to spring, it's very low value. It jumps up in the spring as trees leaf out, and then it kind of falls off very slightly as you go through the summer, and then falls off very quickly when you get to the autumn. Leaves change color, fall off the trees completely, and you go back down to your baseline value. But you might get a lot of outliers in there, depending on what kinds of things are going on in that pixel. So things like disturbances, fires, windstorms, blowing leaves off the trees, things like that. And then you also have variations from year to year in the timing of when things warm up in the spring, how much rainfall you have. If there's a drought in the summer, you may get an early period of the leaves changing color because they're retaining nutrients in their cells. And so you need to take all these into account to figure out how things vary from one year to the next in the climate and how that translates to the way the vegetation is behaving. And that growing season, the time when the leaves are green, determines how much carbon the forests take up. And so if we're going to be able to calibrate things like climate models that have dynamic representations of vegetation, and we want to be able to figure out, okay, how much carbon is this particular area of the forest taking up in a given year? Yes.
Don't mean to interrupt. My apologies for that. We have only, just to make you aware, we have only a few more minutes.
Okay. Actually, I can move on quickly from here. So the second part of this is basically getting the climatology data sets merged with the satellite based data sets. It turned out to be there to look at that 200,000 square kilometers. There are a lot of chunks of data, but each chunk needed to be treated the same way in terms of analysis. And that turned into an ideal task for distributed processing, massively distributed processing. I could do pieces of it at CHTC here at the University of Wisconsin. And then I farmed out a lot of the statistical modeling that you see farther down there in the workflow to the Open Science Grid, which operates basically the same way and we have a great connection with. Things get, it turned out that in my dissertation, I ended up using several million hours to do a lot of this. But you can see here the idea of matching up the climate variables on the right to the satellite images on the left. And I'm not going to let this run too long. But you can see there we go through the stack of climate variables, matching things up. And then, you know, trimming the area, making sure the geography matches up, and then moving on to the next step. There's a step where we need to shuffle things from stacks of dates that have all the variables in them to stacks of variables that have all the dates in them. That's another process that can be done widely parallelized. That's good for a distributed environment. And then there's the matter of chunking the geographical area into smaller pieces. We can't run 40 million pixels of analysis at one time. We need to break that up into smaller chunks. So it turned out that a 16 by 16 pixel chunk worked very nicely with the climatology data sets and in terms of processing time in order to farm that out to processors on the high throughput computing center and the open science grid. And so chunking things into pieces so that there's a very specific data package that goes with the analyses, and then moving on to the statistical modeling component of it. And that's where we actually take the satellite data, match it up with the climate information, and see how they relate to each other using a partial least squares regression approach. This is probably the most detailed aspect of it, of the work that I did for my dissertation. I worked up a rudimentary approach and rudimentary analysis that was good enough to actually finish my dissertation. And then in the time since then, it's been about 3 years now, I've been working off and on while also doing my actual regular postdoc work in revisiting my statistical model, revising the procedures inside of it, revising.
Hey, Matt, I'm really sorry to interrupt you, but this call, so thank you so much for presenting today. This was really great.
And that's the last slide, but that's where I am right now. And I have more work in workflows to do in the future.
That's awesome. I just want to invite anybody who has follow-up questions to Matt or any of our speakers, please reach out directly or pop them in our call document. And you know, we keep editing that for about a week here after the call, and you're welcome to revisit it later. Thanks again. We learned some things today. The most important thing I learned is Bob actually does things on these calls. So thanks for everyone's patience and flexibility. Really appreciate it. If you haven't signed in now, the link's in the chat. And just a reminder, we recorded this call and we will be putting it up soon, and we'll be sending out an email. So today's topic was workflow tools for portable reproducible data analysis. And a super special thanks to our flexible presenters who rolled with our technical difficulties, to Will and Dennis and Matt. So thanks so much. Our next call is on June 10th. Just a note, I put a link for this in the chat. The virtual residency is coming up June 7th through 11th. It's free. There's lots of good information there for people in the researcher-facing space, so go ahead and check it out. So special thanks to the people who worked on the call. Hey, great job, Gladys and Justin, and Christina, Wirwan, Martin, Annalie, Amy, Katya, Bob. I'm Jana Nugent. Please join us in June when hopefully the weather will be nice and it'll be great to see you. Thanks again for your patience, everybody.
Thank you, everyone.
Thank you.
Thank you.